This procedure involves colonization of the murine nasopharynx with Streptococcus pneumoniae and the subsequent extraction of colonizing bacteria, as well as adherent or recruited cells. This is accomplished by first preparing a bacterial inoculum of the S. pneumoniae strain of interest. The live bacteria is then administered intranasally into unanesthetized mice while they are in a restrainer apparatus. After the desired length of colonization, mice are then euthanized and nasal lavages are performed by cannulating their exposed tracheas and flushing out the contents through the nares. Bacterial numbers present in nasal lavages can be quantified using microbiological techniques. Alternatively, immune responses can be assayed using either flow cytometry techniques to phenotype recruited cells or quantitative PCR techniques to measure levels of expressed mRNA of interest. Prior to beginning, you'll need to prepare mouse restrainers for the intranasal colonization portion of the procedure and cannulated needles for the nasal lavages. A mouse restrainer can be prepared simply by cutting off the tapered tip of a 50 milliliter falcon tube. Once you have created the aperture, make sure to use scissors to polish down any sharp ends. For the preparation of cannulated needles, you will need the following: PE20 polyethylene tubing with an inner diameter of 0.38 millimeters, 1 ml syringes capped with 26 and 3/8 gauge beveled needles, a pair of sharp fine iris scissors, and a pair of forceps. Cut the PE20 tubing with the fine scissors into pieces roughly 2.5 centimeters or 1 inch in length, ensuring that each end has a beveled tip. Using the forceps, carefully slide one of these pieces onto the tip of your needle, avoiding puncturing the tubing side. Cannulated needles can be kept in 70% ethanol until needed, and flushed with PBS directly before use. For intranasal colonization of mice with S. pneumoniae, we recommend using a concentration of 10 to the 9